Today we will discuss about glycogenolysis. So it's a breakdown of glycogen into glucose. We have already discussed about glycogenesis, that is generation of glycogen. So both are separate pathway just like glycolysis and gluconeogenesis. So here there will be generation of glycogen from glucose. The site or metabolic organ will be the same where glycogen is stored, that is liver and muscles. The main purpose of hepatic glycogenolysis, that is conversion of hepatic glycogen into glucose, is to maintain the blood glucose concentration within the normal range. So normal blood glucose range is around 70 to 110 milligrams per deciliter. So if blood glucose falls below that level, at that time the stored glycogen starts to break into glucose. Now that process takes place in the liver. Now remember, glycogen is a polysaccharide. It's a kind of homopolysaccharide where several glucose molecules are attached in alpha 1,4 and alpha 1,6 linkage. So breakdown product will be glucose itself. In case of muscle, the main purpose for glycogenolysis is to provide energy for the muscle contraction. So just summary of overall reaction will be something like this. In glycogen, there will be millions of glucose molecules which are attached either in the chain form or in the branch form. So first, all of them will be phosphorylated, and from the free end of the glycogen, one glucose molecule will be released in the form of glucose 1-phosphate. Okay, so suppose if we say a glycogen has around 100 glucose molecules, then first phosphorylase enzyme will act on it. What it will do, it will release one glucose in the form of glucose 1-phosphate. So that released glucose molecule will be phosphorylated. Okay. So name of enzyme will be phosphorylase and one glucose molecule will be released. So remaining 99 glucose molecules will be there in the chain form. So that will be the overall process in glycogenolysis. So let's understand by the cycle form. So this is the structure of glycogen. All these blue dots, yellow as well as the pink dots represents the glucose molecule itself. All these blue dots are in the chain form. This linkage between this pink glucose molecule and this blue glucose molecules is alpha 1,6. The linkage between this pink and yellow glucose molecule is alpha 1,4. And similarly here at rest other places, all these blue glucose molecules are held together by alpha 1,4 linkage. So that is a general structure of glycogen. Now glycogen phosphorylase will start to act. Where it will act? It will act on this free end of glycogen. Okay, it will act on this chain form as well as here in this branch. It will start with the end glucose molecule here and this one. So here what it will do, it will causes breakage in the linkage of these two glucose molecule. Here there is alpha 1,4 linkage, so that will be broken by glycogen phosphorylase, and this separated glucose molecule will be in the phosphorylated form. So the product will be glucose 1-phosphate. Okay, one glucose molecule at a time will be released in the form of glucose 1-phosphate by glycogen phosphorylase. So this process will continue in the chain as well as in this branch. It will continue until where there will be only 3 glucose molecule left in alpha 1,4 linkage. Okay, so this yellow glucose molecules are linked by alpha 1,4 and they are 1, 2 and 3 in number. So glycogen phosphorylase now will stop then there will be action of another enzyme known as the debranching enzyme. So this product is also known as the limit dextrin. It's a breakdown product of glycogen itself. This debranching enzyme, what it will do, now it will transfer these three glucose molecules into the chain. Okay, so basically they are removing the branch of glycogen, so that's why the name is debranching enzyme. So these three glucose molecule now will be shifted from its branch to the main chain. Okay, so you can see here these three glucose molecule now in the chain form, and one glucose molecule will remain, which is there in alpha 1,6 linkage with this blue glucose molecule. Fine. So now this debranching enzyme will act on alpha 1,6 linkage here, and it will release this glucose molecule. Now the only change here, if there is a breakdown by glycogen phosphorylase, the released form of glucose is glucose 1-phosphate. In case of debranching enzyme, the released will be the free glucose. Okay, so that is the difference in action of glycogen phosphorylase and debranching enzyme. Further on, now we don't have any branch here, and all these glucose molecules are in alpha 1,4 linkage. So glycogen phosphorylase once again will start to act in individual branch, and here there will be loss of glucose molecule in the form of glucose 1-phosphate. Okay. So it's a mechanism which takes help of only two enzymes, glycogen phosphorylase and debranching. Okay, and the end product will be glucose 1-phosphate molecules mainly, and after 10 to 12 steps the free glucose molecule will be released. Okay, so in glycogenolysis the end product was glucose 1-phosphate, but our main aim is to produce the free glucose. So from glucose 1-phosphate there will be formation of glucose 6-phosphate. Enzyme name will be phosphoglucomutase. We have already discussed that enzyme in gluconeogenesis as well as in glycogenesis. Okay, so in glycogenesis we have already learned conversion of glucose 1-phosphate to glucose 6-phosphate by phosphoglucomutase, and this glucose 6-phosphate will be released in the form of glucose with the help of enzyme glucose 6-phosphatase. So the main point here, if glucose 6-phosphatase is there, then a tissue can contribute towards the free glucose. Otherwise, the product of glycogenolysis will be glucose 6-phosphate, not glucose. So that process makes liver glycogenolysis and muscle glycogenolysis differ from each other. Liver has presence of glucose 6-phosphatase, so it will contribute towards the blood glucose when the supply of glucose or blood level of glucose starts to fall. In contrast, in muscle there is an absence of glucose 6-phosphatase. So in muscle glycogenolysis, end product will be glucose 6-phosphate, which undergoes the process of glycolysis and can generate the ATP for the muscle contraction. So please note down that difference of muscle glycogenolysis as well as liver glycogenolysis. So muscle cannot contribute towards the maintenance of blood glucose in the blood. Okay, fine. Now coming to the regulation part. So the activator of glycogen phosphorylase will be mainly AMP. The end product mostly will inhibit it. So if we have sufficient amount of ATP or glucose 6-phosphate, then they will inhibit the glycogen phosphorylase, because if we have sufficient amount of energy, then there is no need to production of glucose itself. Now coming to the hormonal regulation, as we already know that insulin will inhibit those pathway which generates the glucose. So in glycogenolysis there is a production of glucose, so insulin will inhibit it. So here the regulatory enzyme is glycogen phosphorylase. It has two forms. phosphorylated form and dephosphorylated form. Here the phosphorylated form is the active form. So if there is addition of phosphate to glycogen phosphorylase, that means it is active. If there is a removal of phosphate, then it is inactive. So by our common funda, insulin, which inhibits the glucose generating pathway, what it will do? It will activate the enzyme protein phosphatase. So the protein phosphatase will be active. It will remove the phosphate from glycogen phosphorylase. So enzyme will become inactive, and then there will be no formation of glucose 1-phosphate from glycogen. You have to see the right side of the image itself. Left side represents the glycogenolysis. So that is the action of insulin. In case of glucagon and epinephrine, what they will do? They are hyperglycemic hormone. So they will stimulate the formation of glucose 1-phosphate from glycogen. So they will stimulate the glycogen phosphorylase. An active form of glycogen phosphorylase is by addition of phosphate. So they will stimulate the enzyme phosphorylase kinase by increasing the cyclic AMP production. Okay. So in that fashion, glucagon will stimulate the glycogen phosphorylase and will stimulate the glycogen breakdown, that is glycogenolysis. Okay. So insulin will inhibit glycogenolysis. In contrast, glucagon will stimulate it. Now there are certain disorders related with the glycogen metabolism. They are known as the glycogen storage disease. You should remember the name of the disease as well as the deficient enzyme present in it. So type 1 glycogen storage disease is known as the von Gurk's disease. It is the commonest of glycogen storage disease, and here there will be deficiency of enzyme glucose 6-phosphatase. Second is the POMS disease. Here there will be deficiency of enzyme lysosomal maltase or glycosidase. Third is the Coris disease, where there will be deficiency of enzyme debranching. Okay, we already seen debranching enzyme in glycogenolysis. Another one is the fourth is the Anderson disease, where there will be deficiency of branching enzyme. That was the enzyme present during the glycogenesis. Fifth one is McArdle's disease. Here the deficiency of enzyme is muscle phosphorylase. And sixth one is the Hirsch disease. Liver phosphorylase is the deficient enzyme. So these are the 6 types of glycogen storage disease. Ideally, you should remember the names and the deficient enzyme. In detail, the important one out of this is only the type 1, that is von Gurk's disease, when there will be deficiency of glucose 6-phosphatase. So in von Gurk's disease, it is the commonest type of glycogen storage disease. Deficiency of enzyme will be glucose 6-phosphatase. So glucose 6-phosphatase, if it is absent, then there will be no conversion of glucose 6-phosphate to glucose. Free glucose will not be there. So this glucose 6-phosphate will be accumulated and it can generate the glycogen. We have already seen in the case of glycogenesis. So this accumulated glycogen can lead to enlargement of liver also. So that is one of the finding in case of von Gurk's disease, hepatomegaly. Another one is the hypoglycemia. Why there will be hypoglycemia? Because free glucose is not available. Okay. If glucose 6-phosphatase is not active, no free glucose in the blood. So patient will become hypoglycemic. Another thing is the lactic acidosis. Why? Because if there is more glucose 6-phosphate, it can go under process of glycolysis, and since there will be also chances of anaerobic glycolysis, so accumulation of lactate will be there and there will be lactic acidosis. Similarly, accumulation of acetyl-CoA will lead to hyperlipidemia as well as the ketosis that you will learn in lipid metabolism. In hyperuricemia is also there, that we will discuss why accumulation of this glucose 6-phosphate leads to hyperuricemia in our next class, that is of HMP shunt. Okay, fine. So if there is a question like glycogen metabolism, then you have to write both the things, glycogenesis, that is the generation of glycogen, and glycogenolysis, that is the breakdown of glycogen. Okay, if there is a separate question on individual pathway, then you have to write only that pathway itself. Okay, so that was about the glycogenolysis. Thank you.